Hi folks, welcome to the third and final day of the Toronto workshop on reproducibility. Today we start off with Professor Marco Antonio Maximo Prado, who is Canada Research Chair in Neurochemistry of Dementia and a scientist at the Robarts Research Institute. So very interested for his topic today, which is reproducibility for behavioral experiments in basic science. So thank you very much, Marco, for joining us.
Well, thank you for the invitation, and it's a great initiative to discuss reproducibility practices, best practices, and different sets of research. I'm going to talk to you about some of the efforts that we are driving here at Western, and this is really part of a very large group. I'm just representing a number of people here, and the goal here is really to try to improve the way that preclinical science or basic science is done, and particular work with animal models. And this graphs just give you an idea of the amount of time, effort, and work that takes to develop the drug. And particularly for drugs in the nervous system, this is probably even a little bit longer and worse. So you start with a lot of compounds. You try to find a target. You validate that target. Eventually, you get to this point where it's what we call the preclinical phase, where you have a number of drugs to be tested. And you can see this takes quite a bit of years. And you're making sure that whatever compound you have is effective, is safe, and then that would lead for an investigative drug trial here in humans. So the question is that in particular in dementia and in Alzheimer's disease, there's really not been any new drug approved besides an antibody against amyloid beta last year that was very, very controversial and is not approved in Canada. And one of the reasons for that, there's a big, what we call the valley of death here, where work here doesn't necessarily translate to clinical trials. So things that are sometimes found effective here, they are not found effective in clinical trials. And one of the goals of what I'm going to show you is to try to improve that. And so the problem is how to predict which drugs will improve cognitive symptoms. And I can tell you, there is plenty of stories, and there is for example, there is a company now, and I'm not going to even mention the name, that's receiving a lot of controversy because of some of the science they did with the compound that they are driving for clinical trials is really not to the standard that one would like. And the company has been shortened in the stocks, and there's been a lot of controversy. So we really want to have as much vigor and reproducibility in this side here as well as in the clinical phase. So for that, you need animal models, and the top animal model for this type of research is mice. And I've just put here, I really love this sort of slide because it shows you all the different types of science that have been advanced, including the number of Nobel Prizes that have been advanced by using mice as a model. And in terms of the research that I do and that we're interested is in neurodegenerative diseases first, but obviously we're interested in any disease of the brain. But I just want to briefly explain how we model that in animal models. So Alzheimer's disease, as you know, is a combination of genetic and lifestyle factors that increase your risk. Most of the cases are sporadic, but there are a few cases that are genetic. There is a number of hallmarks that you see in terms of pathology of Alzheimer's disease. So neurons stop communicating with each other, chemical messengers or neurotransmitters dysfunction, particular one named acetylcholine, but others as well. Then you have accumulation of the so-called plaques and tangles, which are pathology hallmarks. The brain gets inflamed as well during this process. And this is happening for probably 20 to 30 years before people actually develop the sort of cognitive dysfunction that led you to go to a doctor and do some testing and then get diagnosed. And by the way, diagnosis of Alzheimer's is always right now is basically a cognitive testing that's not probably the best way do it. And so this is a type of pathology that you see, that's your brain shrinks, when you have this pathology in Alzheimer's. And I just want to, I'm not going to go into detail here, but it's just to show you that what happens here is you accumulate a protein called amyloid beta, which is shown here. And that comes from another protein named amyloid precursor protein. And genetic cause of Alzheimer's disease, the gene for this protein called APP, or genes that code for enzymes that process amyloid beta, they have mutations, and those mutations lead to Alzheimer's disease. So in mice, what we do is basically we reproduce those mutations to have mice that have the same sort of pathology that you'd see in Alzheimer's disease. But the problem is when you go test the major complaints of patients, right? So if you have Alzheimer's disease, your major complaint is that you really can't remember where things are, if you left with your car or not, you start wondering. So you test this in mice with cognition and behavior testing. So the problem here is that if you look at this sort of table, and this is from my review from a few years ago, and there's different mouse models of Alzheimer's disease right here, all generated using genetics, like I just mentioned, mutations on this gene called APP. But if you look at the behavior, it's really variable. And not only that, it's very variable. So different labs, sometimes they find effects, sometimes they don't find effects. But also this behavior is not really similar to what you do in humans. So for example, in mice, one of the top ways to test for memory is throwing a mouse in a pool of water and asking the mouse to find its way out. So you don't get Alzheimer's patients and throw in a swimming pool and ask them to go and find their way out, right? There's a huge component of stress, and that affects the mice. And there's other types of tests that you do in mice, and most of them are not reproducible. So enter now, what the problem is. So you want to test behavior in mice in models of dementia, but the dysfunction you see in humans affects the activities of daily living, affects how you live your day-to-day. So how are you going to ask those questions in mice? The second ideally way to do this behavior tasks is that you want to have something that is somewhat similar to what humans do, that involve the same sort of brain regions and involve the same sort of neurotransmitters. So if you find drugs that are good for the mice, they might be also working for humans. You want something that's reproducible. And I just showed you that a lot of these tasks are hand, people just use a stopwatch and record by hand. So you want something that's automated, that can be standardized as well. But also you want throughput. And the sort of testing that we do, we actually test about 1,000 mice a day, every day. And that allows us to test multiple drugs or multiple mouse models. So this comes from the work from two colleagues of mine that started this almost 30 years ago. Tim Bussey and Lisa Saxida, they were in Cambridge for quite a bit of time, and they moved to Western about 5 years ago. And they developed a series of cognitive tests for animal models that use touchscreens, similar to iPads for mice. And they've shown over the years, they validate those tests. There's a number of those tests, they are validated. And they show over the years that, humans and mice with the same mutations or same disease, they have somewhat similar cognitive deficits. And sometimes the task that you use in the mice is very, very similar, if not identical, to the tasks used in humans. So there is about 30 different validated tests that test multiple cognitive domains, all using the same sort of apparatus, which is a bit of an advantage. So you can really do what you do in humans. So instead of doing this one task, you test a battery of things in the mice and look for the cognitive domains that are affected. So just I'm going to give you one short example. This is a task of, we're asking the mice to tell us if it is paying attention or not. And the way we do this is that's the apparatus. There's 5 screens. The mice is going to see a flash of light. We're not going to see any flash of light. And if the mice gets the right screen, it gets rewarded. If it gets the wrong screen, it doesn't get the reward. And the reward is given here at this point, and it's really nice Canadian strawberry milkshake. So let's see what the mouse does. So it starts by touching this, and that triggers the computer. So the computer is monitoring everything through infrared. So he gets the right screen, gets the reward. Between one, that's my student when he was seeing this for the first time, actually. So between one test, the other one, there's an intertrial interval. The mouse knows it doesn't have to do anything until it starts again by touching the receptacle and getting a cue from the computer. And you can see that, when it starts, there we go, gets a cue, mouse turns around, it's looking for the screen. We don't see the flash, but it's very fast. And then it gets. So when we do this, what you do, basically, you force the mice to tell you if they're paying attention or not by measuring the number of correct answers or missed answers. There is a number of parameters that we can measure. And you tax attention by decreasing the time that the flash of light shows up from 1.5 to 0.4, 0.2 seconds. And you can see that, if you tax attention, the accuracy or the right answers decrease as you'd expect. And this is just one test that we did. So these are experiments that were done both here at the University of Guelph with the same tasks. And you can see that we're comparing, normal mice doing this task. We compared Alzheimer's mice as well doing this task. And you can see that it's really not much difference between the two sites. We test this in male and females. In fact, if we compare all the parameters that we can measure in mice with this and other tasks, 85% of the parameters that we find are essentially identical between two different sites. And not only that, we reproduced the data that had been published before. So this is really reproducible. And that's another mouse line that we tested as well. So using this, our group has built what we call cognitive platforms. And we call cognitive platforms a combination of the correct mouse model and the right cognitive testing that provides an answer whether there is cognitive deficits or not, and that can be rescued by drugs or by treatments. So you can have an idea that, we tested actually quite a bit of different mice. We tested male and female mice. I'm not going to get into details. We test different cognitive domains, and a lot of these are ready now, and they've been used. And we have been able to show that if you rescue some of the toxic protein misfolding, you can actually rescue the cognitive deficits. This sort of analysis also is great because with the touch screens, we can use a lot of neurotechnology. For example, in humans, you could look at using MRI, for example, to look at changes in brain regions. You cannot do that in mice, obviously. You cannot do the mice. I mean, you can use MRI, but you're not with the mice doing this behavior test. But what you can do is to record neuronal activity. And we use something called fiber photometry, where we put fiber optics in the brain of the mice. And we can record, for example, calcium signaling from different genetic defined neurons. We can record, for example, neurotransmitters. In this case, we use a sensor for the neurotransmitter acetylcholine. That's important in Alzheimer's disease. This has been published a couple of years ago with our collaborator from China, Yulong Li, who developed those sensors. And you can see that when mice are performing the touchscreen, you can actually record at millisecond resolution changes in acetylcholine. For example, when they get a reward, there's this huge decrease in acetylcholine signaling in the brain. And we know this is specific because we have mice that release very little acetylcholine, and you don't see those changes in acetylcholine being secreted in the brain. So you can collect a lot of information. You obviously could collect structure MRI and function MRI in the mice, anesthetize, but not doing the task. You do these cognitive tasks. So the question now is what you do with all this data, how you make this data available to increase reproducibility, increase views. So we have two open science initiatives, and they are really run by a number of people, including my colleague Tim Bussey, who takes care of a lot of this. One of them shares knowledge on use of touchscreens. It's called touchscreencognition.org, and it's basically your Facebook for this sort of tests where people can post their papers, they can discuss, we have a lot of SOPs already there. There are videos that people can access of training, etc. And there's about 400 labs in the world using this sort of technology. And we also have a data sharing And I'm going to focus most of the data sharing, which is what we call it a MouseBytes, of the MouseBytes project. And you can access this through www.mousebytes.ca. So this has been published a couple of years ago in eLife, and the information there, if you were interested. A lot of people have been involved in doing this. So this is what is the front end when you access MouseBytes, and I'm not going to have time to go over all the different features, but you can see that right now we have about almost 3,000 mice. There's a little bit more now. This is from a couple of weeks ago. About 3,000 mice registered, where we have the cognitive performance of those individual mice deposit in a structured way with metadata that anybody can go and download. There's a number of features in this website and database. So you can download touchscreen data, you can upload touchscreen data. We have MouseBytes Plus or complementary data. For example, you can link fiber photometry or MRI data to your cognition through this secondary database. There's analytics, there's really a number of things. And not only that, we use typical Spotfire. And so you can actually visualize the performance of the mice on the fly. So you can ask the program to basically build graphs for you, and you can see these graphs in real time. So this again is the architecture of this database. I'm not going to get into the details. Uploading data, you do have to be registered. So integrity of the data depends on the PI. So we link to the PI, and this not only is available on a website to be extracted, both the complementary data or MouseBytes Plus, but all the structured behavior data. But you also can access this through CONP as well, and it's all Creative Commons. Anybody can download and use. You can also create specific unique links for data sets. So when you publish a paper and you have data that goes to a graph, you can create a link that allows you to pull the data from that particular graph or panel. This is the number of people around the world that have access or are users of MouseBytes, different countries. Just to give an idea that, obviously this started a few years ago. It takes a while, and we're still trying to convince a lot of people to deposit their data in the database, but some already have been convinced. We do follow the FAIR principles and try to work within those the FAIR principles and compliance, and we collect both for the complementary data as well the structured behavior data, a lot of metadata that can be used. So what does that take us? So we create this database, and we're depositing this database. So we're running hundreds of mice on this platform. So what can we do with this? So one of the things that we created, or we're starting to create right now, is an initiative with colleagues at McGill. We call it Trident. Translation Initiative to De-risk Neurotherapeutics. This is being led right now by one of my colleagues, Rav Menon, who is an expert in imaging. And it has, obviously, it's a multi-PI led platform. And it's really thinking about different ways to develop preclinical research to facilitate discovery that it will be valid for clinical trials. So we have platforms for patient-derived brain organoids. So there's organoids that you do in vitro and basically using cells from humans to generate human cells that you can use for screening different drugs. If the drugs are, get to have good effects here, we can move in mice, and we have this, it's not pathology that we're using, it's both biomarkers for imaging and cognition. So pathology is really an endpoint. In Alzheimer's, there's plaques and tangles. So we're trying to see toxicity that we can detect either by using imaging biomarkers or by using cognition. And as I mentioned to you, there is really a number of cognitive tests in mouse models and combination of mice and cognitive testing. If the drugs pass this part here, and we do this in a multi-center way, so we're doing this both at Western and at McGill at the same time. So it's somewhat similar to clinical trials. We are, the idea is to have partnerships with industry, but being arm's length. So we really don't have any financial or other interest that a drug will succeed or fail. In fact, if we fail, we want to fail fast, right here in the first stage, either in organoids or in mice. And if the drug passes this, or compound or gene therapy past those two, then we can move to marmosets. And now we're developing different marmosets models that mimic the mice and also use the same sort of cognition and same sort of imaging. So the combination here is, we try to use humanized cells, but also a lot of the mice we use are humanized as well. And then marmosets obviously are very close, non-human primates, so they're close to humans in terms of genes and also brain. And we use also biomarkers of cognition and imaging that are used in humans. And all of this is doing under open science, an open science framework. So the data becomes available after published. And the idea is that we would be able to help to predict, to better predict which drugs might be successful in clinical trials. The combination of touchscreens and imaging, a number of imagings and using deep learning and biased aligners for imaging, and a number of those biomarkers are being developed with my colleague, Mala Chakravarti at McGill University, and as well with people at Robarts. So, the idea is really that we think that working together instead of competing and making this a large consortium, will have a better chance to help to address this really important gaps in drug discovery and help to make the basic preclinical research in animal models that we think it's a huge problem right now. And I don't say we have a reproducibility crisis. I never liked that comment. I really think that science corrects itself, but we have to strive to be as rigorous as possible with the public funding that we receive. And the idea is now, really making everything that we do in terms of SOPs, tasks, as rigorous, but as well as open and transparent as possible. So what's a measure of success? So obviously we really want to make sure that, if we're doing things in 2 different sites, we find, the same things in both sites. Otherwise we're not doing a good job. We hope that in the future, there'll be crowdsourcing and reuse of data. So for example, we have one person interested right now using this database to compare male and female mice. And I don't think you would ever be able to compare it. So we probably have a little more male than female mice in the database, but let's say we have at least 1,000 of each. So I don't think you've ever been able to compare 1,000 mice in different cognitive tasks and compare sex. You can compare mouse strains, you can compare different genetic manipulations, whatever that you want, that the data is available. So obviously increased access to this open data that people can reuse. We hope to also contribute to better understanding of the circuits that are involved in cognition and provide these platforms for drug testing that would be validated and maybe disseminated to other universities and other groups as well, and obviously collaborate with industry. So ultimately, the goal is to really synergize efforts to improve our preclinical testing. And one of the things that we hope to be able to do is things that pass to our rigorous testing at the clinical level will get at the preclinical level will be successful in the clinical testing, and that we won't only find out in a few years. Although we are testing some drugs that have previously been successful before in preclinical testing and fail in the clinical to see if we can find they fail first. All right, so I'm just going to finish by thanking the people that have been involved in this, and really a number of people to thank, but I was just going to mention the top ones. A lot of this open science initiative is being done in collaboration with my colleague, Tim Bussey. Work in my lab is also directed by my partner, Vania Prado. A lot of what we've done was driven by Flavio Beraldo, who left for industry now. But now a lot of the software part, or actually most of the software, if not all of it, was done by Sarah Memor, who's still with us and still working at MouseBytes. So I'm gonna just mention that a lot of this has been done through funding through our CFRAS BrainScan, some early funding from the Western Brain Institute and Brain Canada as well. But BrainScan has been really supportive of this sort of initiative. And I'll be happy to answer any questions if you have. Thank you.
Thank you very much, Marco. I really appreciate your presentation. Does anyone have any questions or comments? You can just raise your hand. I'll just start with my understanding is that NSERC is changing some of the requirements around data sharing and reproducibility. Does that affect you, or you've sort of got ahead of it with some of these initiatives?
Yeah, I think we got way ahead of it with the initiatives. In fact, that's a really good question. I tell you that in all my grants, all the grants that I write, both to NSERC and CHR, that use touchscreens, we always have the statement that the data, and in fact, we do that, the data are going to be shared. And in general, we receive, it hasn't been, I don't think it has yet permeated to the institution, but reviewers have been very supportive. They really like the fact that all the data are going to be shared and be open and could be reused.
Yeah, that was one of the experiences of Jay, who was talking yesterday in material science. Also, he found that reviewers were increasingly positive about that. When did you see that change sort of happening in your field?
Yeah, so I'm a molecular neuroscientist, and so I've been trained in doing behavior in mice after I became a professor. So being a molecular neuroscientist, I was really bugged by the fact that there was a lot of variation between labs of the behavior. And so that started to bug me. So we started this project actually in 2013. It took a few years to fly. Obviously, it's a huge effort. But I think, I mean, the ties are really changing now. I think that it's wonderful to see so many people committed. That being said, I think that it's important to not minimize the amount of extra work and extra support that one needs for data management, right? So that I think is going to be something, and I know CHR is looking at this, and probably in NSERC as well.
And final question, I think one of the countries that stood out, I mean, Canada, obviously there was on your map that you shared, Canada, obviously there was a lot, the US, there was a lot, but was there Indonesia also?
Well, yeah, I mean, these are people that have
right.
access. So what my colleague Chimbase does, he leads this, or this touchscreencognition.org, is one of the things is running a touchscreen symposium every year that was in person but then became online for the last 2 years, obviously. But one of the things that we were lucky to get some support and bring people from all over the world in the first year. So we had about 20 to 30 trainees that we could bring from different countries, including people from Indonesia. We had people from China, from Brazil, from really a lot of support for developing countries that probably are new to the technology.
Well, thank you very much for a very interesting presentation, Marco.
No problems, my pleasure. Thank you.